In some of the previous videos, we had learnt that the characters are carried from one generation to another generation through genes. The genes are carried on chromosomes. Mutation is a change. These changes are carried from one generation to another generation. The term mutation was introduced by Hugo de Vries in 1901. Hugo de Vries introduced the term mutation based on his observation on the plant Enothera lamakiana. This plant is called as evening prime rose. This plant opens the flowers after sunset. Hugo de Vries observed in these plants with large size flowers and leaves in some of the plants. Some of the evening prime rose plants produced large size leaves and flowers. He mentioned these changes as mutation. Mutation is sudden change in the genetic material, that is sudden change in DNA. These changes are heritable changes. That means these changes are carried from one generation to another generation. There are two types of mutation. One is chromosomal mutation, another one gene or point mutation. Chromosomal mutation are of two types: change in the structure of chromosomes and change in the number of chromosomes. What is chromosomal mutation? Sudden change in the structure or number of chromosomes is called as chromosomal mutation. These changes are called as sudden change because these changes happen in one generation. And changes in the structure of chromosomes is a kind of chromosomal mutation. During cell divisions, sometimes the chromosomes break and rejoin. This results in change in the structure of chromosomes. Chromosomal mutation may be change in the structure of chromosome and change in the number of chromosome. Change in the structure of chromosome is due to chromosomal breakage and rejoining. In this diagram we see a breakage in the chromosome. There is loss of a part of chromosome. The gene A is lost. This type of changes are called as deletions. Sometimes the damaged part of the chromosome joins into another chromosome. In that case, the chromosomes have repeated genes. Here the gene B and C repeats. This type of changes are called as duplications. In this case, the order of genes is reversed. D, E, F is reversed. This type of chromosomal changes are called as inversions. Here the order of genes is A, B, C, D, E, F. After F there is no G. GHI is on another chromosome. GHI is exchanged with RST. This type of exchange of exchange of genes between chromosomes. GHI genes are exchanged with RST genes. This type of exchange of genes between chromosomes is called as translocations. Deletion, duplication, inversion, translocations are changes in the structure of chromosomes. Changes in the number of chromosomes, that may be increase in the number of chromosomes or decrease in the number of chromosomes. Increase or decrease in the number of chromosomes is also called as ploidy. There are two types of ploidy. They are euploidy and aneuploidy. We have two sets of chromosomes in the body cells. 23 chromosomes form one set another 23 chromosomes form another set. So we have two sets of chromosomes. The total number of chromosomes is 46. When there is change in the sets of chromosomes, we have two sets of chromosomes in our body cells. Changes in the form of sets of chromosomes is called as euploidy. Euploidy is nothing but change in the number of sets of chromosomes. Aneuploidy involves addition or decrease of one or more chromosomes within a set. One or chromosomes are changed in one set or two sets. Here sets are increased, here chromosomes are increased or decreased. What is euploidy? Usual number of chromosomes is diploidy. What is usual number? Two sets, n plus n, that is diploid. Change from diploid condition is called as euploidy. 2n has n plus n, two sets of chromosome. When there is addition of one more set, that is 2n plus one more n, 3n, that is triploidy. If an individual has three haploid sets of chromosomes, is called as triploidy. Haploid is single set. When there are four haploid sets, that is tetraploidy. Here the triploid individuals have no seeds. Banana has seeds, that is diploid, two sets, normal, that is not cultivated. Cultivated banana has no seeds, that is example for the triploidy. Triploid individuals are sterile, they do not produce seeds. Presence of four sets of chromosomes is called as tetraploidy. Tetraploid plants produce large size fruits and flowers. Tetraploidy increases the size of fruit and flowers. So tetraploidy condition is commercially important. Triploidy and tetraploidy are commonly called as polyploidy. What is aneuploidy? It is a loss or gain of one or more chromosomes in a set. If there is loss of one chromosome, we have 46 chromosomes, 2n is 46, so 46 minus 1, 45, that is monosomy. In a particular pair, one is removed. Addition of one chromosome, trisomy. In a particular place, there are three chromosomes, so the total will increase. When there is loss of pair of chromosomes, that is nullisomy, 2n minus 2. So trisomy condition in human results in a disease, that is Down syndrome. Down syndrome is an example for aneuploidy. The particular aneuploidy of Down syndrome is trisomy. The Down syndrome individuals have poor mental capacity. This is a karyotype of Down syndrome. Here there is abnormality in chromosome number 21, extra copy of chromosome number 21. There are three chromosomes in the 21st. This is called a trisomy 21. They have mental retardation, they have poor mental capacity, and they have delayed development. Growth is slow, they are short. The individuals with Down syndrome have behavioral problem, and they have poor muscle tone. And the stiffness of muscles at rest is called as muscle tone. And they have problem with vision and hearing. Down syndrome is called as trisomy 21. Gene or point mutation, that is change in the structure of gene. A chromosome has important component DNA. The DNA has several genes. In human cells, the DNA has more than 30,000 genes. Changes occurring in nucleotide sequence of a gene is called as gene mutation. What is nucleotide? DNA is made up of nucleotides. There are 4 nucleotides, They are adenine, guanine, thymine, cytosine. What type of changes are possible? Substitution. If A is replaced by another or G is replaced by another, that is substitution. Gene mutation involves substitution, deletion, and insertion. Loss of one gene, loss of one nucleotide in a gene, that is loss of A or loss of G, that is deletion. When one more nucleotide is added, that is insertion. When there is extra A or extra G, that is insertion. So these types of changes in gene results in unusual protein. A gene carries information for a protein. When there is mutation in the gene, that will produce unusual protein. Gene mutation results in abnormal protein. Genes occupy particular place or particular point in a chromosome. So gene mutation is called a point mutation. This abnormal protein formation results in disease. Sickle cell anemia is caused by the mutation of single gene. Normal gene produces normal protein, that is normal hemoglobin. When the hemoglobin is normal, the RBC is normal. What is the shape of normal RBC? Circular biconcave. Because of mutation in a single gene, that is change in single nucleotide, that results in abnormal protein, abnormal hemoglobin, that makes the RBC sickle shaped. This sickle shaped RBC cannot pass through small blood vessels, that results in serious conditions. So gene or point mutation changes new protein and changes the phenotype. Sometimes this kind of changes results in evolution also.